Hi, my name is Teisha King, and I'm a graduate student at Louisiana State University. And today I'll be discussing behavior and neural expression of non-redundant signaling during courtship in an African cichlid fish. So during courtship, many animals use various types of displays to attract mates. And in order to ensure that the signal is received, the sender often sends information through multiple sensory channels, which can be redundant and supply the same information to the receiver and therefore strengthening the original signal, or non-redundant, where the added signal supplies new information to the sender. And an example of redundant signaling is seen in some species of moths, where females have the same behavioral response to pheromones and sounds produced by males. And an example of non-redundant signaling is seen in tropical wandering spiders, where females release hormones to attract males' attention, which is followed by vibrations that they use to orient males towards them. So while many studies focus on the output of multimodal courtship, few studies focus on how these signals are processed by the receiver. In the African cichlid burtoni, dominant males are brightly colored and perform body quivers during courtship displays. And these quivers are also produced in conjunction with low frequency sounds, which make burtoni a great model to study how multimodal audio and visual reproductive cues are processed by females. And here I just have a quick video to show an example of what the quiver looks like in conjunction with the low frequency sound. So today, the two questions I'll be focusing on are: one, does male visual acoustic courtship signaling convey redundant or non-redundant information? And two, where are unimodal and multimodal visual acoustic signals processed in the female brain? In order to explore how auditory and visual signals are processed by females, we manipulated barrier type to control reproductive signals delivered from dominant males. And I also wanted to point out the association zone that I have marked off here at the bottom. And this is what we use to quantify female affiliation in conjunction with the time that the female spends at the barrier. So for control trials or the control condition, there was no dominant male present, so the female didn't receive any visual or auditory cues. For the auditory only, there's a black barrier separating the subject female from the community, so the female can hear the low frequency sounds without seeing any of the quivers. For the visual only, there's an airtight seal separating the community from the subject female, so the female can see the visual cues being the quiver without hearing any of the low frequency sounds. And finally, for our last audio-visual condition, the female can see and hear reproductive cues produced by the dominant male. For the timeline of the methods, we first started off with a 10-minute acclimation period where we have a blue acrylic barrier separating the community from the subject female. The blue barrier is removed and we quantified or recorded behavior for 30 minutes. Next, we quantified male courtship behavior and female affiliation, which is the time she spends in the association zone and the time that the female spends at the barrier. Next, we performed in situ hybridization for cfos and immediate early gene, which we use as a marker for neural activation. And finally, we quantified neural activation in nuclei of the social decision-making network, which are a collection of nuclei that are conserved across vertebrates and thought to be involved in decision making. So for our results, first here we have a graph that shows the total male quivers that were produced in auditory, visual, and audio-visual trials. And we see that there's no difference in the amount of quivers that males produce. So this shows us that male effort is the same. The only difference is in the stimulus that the female receives. Next, we have two graphs showing the time that females spend in the association zone and at the barrier. And we can see that for auditory only trials, females spend significantly less time in the association zone and at the barrier. So because female response is different when she receives only auditory cues, this tells us that the auditory and visual reproductive signals are non-redundant. Next, when we take a look at the brain activation data, here we have the number of cfos positive cells for the ATN, and we can see that females exposed to visual or audiovisual signals from males showed a greater activation than females exposed to auditory cues or no male signals at all. And this makes sense because the ATN is known to receive both auditory and visual inputs and also plays a role in reproductive behaviors. And this is in contrast to other regions that we quantified, like the caudal VV, where we didn't see any differences in neural activation across conditions. And next we have our results from a principal component analysis where we input all activation data across all 4 conditions. And we identified 3 distinct neural activation networks. The first here in green, which we believe likely represents regions that process unimodal information, so either visual or auditory alone. The next region here, or next network here in blue, which we believe process multimodal visual and acoustic information because it was characterized to have high activation in audio. These regions were characterized to have high activation in audiovisual trials. And finally, our last network here shown in yellow, which we believe are areas involved in any processing any social input, because they have similar activation patterns across all 4 of our experimental conditions. So finally, to conclude, today I've shown that auditory and visual courtship signals from dominant males function as non-redundant signals, and that the visual component is dominating the auditory, and that there are differential activation patterns between each of the conditions that we explored today: control, auditory, visual, and audiovisual. Moving forward, we would like to further explore the relationship between the neural activation patterns, male courtship, and female affiliation, and also look at some neural recordings to investigate the electrophysical properties of nuclei in the female brain that are responsible for processing multimodal cues. And with that, I would like to thank the Maruska Lab and specifically Brandon and Emily for helping to collect data for this project. And with that, I'll take any questions. Thank you.